video on the unit on bacterial metabolism. In the past videos, we've been following the breakdown of glucose as it gets oxidized all the way to carbon dioxide through the pathways of glycolysis, pyruvate oxidation, and the TCA cycle. So if you remember, by the end of the TCA cycle, we were left with 10 molecules of NADH and 2 molecules of FADH2. Those electron carriers now need to go drop their electrons off at the electron transport chain for the last stage of respiration. So let's take a look at how electron transport systems actually function.
The electron transport chain, or electron transport system, is the last stage of respiration. It consists of a series of membrane-bound proteins that all have an increasing affinity for electrons as you go down the chain. This means that each protein in the chain actually wants to bind the electrons a little bit more than the protein that was before it. So it's going to steal the electrons from it and pass the electrons from one protein to another. As these electrons get passed, they're going to release a little bit of energy, and that energy is going to power the transport of hydrogen ions out of the cell. It's important to remember that there are no mitochondria here. The proteins of the electron transport chain are actually embedded in the bacterial cell membrane, not in a separate compartment. So in order to continue passing electrons, the electrons are going to be removed at the end of the protein chain by an inorganic molecule called the terminal electron acceptor. This is the only conceptual difference between aerobic respiration and anaerobic respiration. In aerobic respiration, that terminal electron acceptor is oxygen, while in anaerobic respiration, it's something else. Species often use things like sulfur. Or others like nitrate or sulfate. The overall purpose of the electron transport system is to generate the proton motive force of hydrogen ions. This proton gradient can then be used to power ATP synthase enzymes to make ATP through a process called oxidative phosphorylation. Next slide, we're going to take a look at what this actually looks like. Okay, so here we have our bacterial cell. So this is the normal cell membrane. And embedded in that cell membrane are the proteins of the electron transport chain. Also embedded in the membrane is a protein for your ATP synthase. This process starts when your NADH that has already been made in those previous steps of respiration comes along and drops off electrons to the electron transport chain. They drop them off at this first complex 1, so the first protein in the chain. And when those electrons are dropped off, NADH is now oxidized back to NAD+, which remember can now go back to things like glycolysis or the TCA cycle to pick up more electrons and come back and start the process again. Once the protein has the electron, those electrons are then passed all the way down the chain because each protein in line wants those electrons more than the first one, so it pulls them away. So here we have step 1, electrons are dropped off. Step 2, electrons are transferred. And then during that transfer of electrons, the energy that's being released from the electrons is going to be used to power the pumping of protons out of the cell. This creates a proton gradient, what we also call the proton motive force, where we now have a high concentration of protons outside the cell compared to a lower concentration in the cytoplasm. Anything that has high concentration on one side and low concentration another will like to diffuse down its gradient. And so those protons will diffuse through ATP synthase, and that's going to power ATP synthase to take ADP plus inorganic phosphate and combine them to make ATP. So step 3 of the electron transport chain was the pumping of the hydrogen ions to create this proton motive force. Now the last step is that this electron that's here sitting in this last protein needs to be removed from the electron transport chain. These proteins can only hold so many electrons, so if this one stays here, then we can't pass any more down and the whole chain gets backed up. This is where the difference between aerobic and anaerobic respiration comes in. If this is aerobic respiration, then what is going to pick up the electrons at the end is oxygen. And that's going to form water, which is a byproduct of respiration when you're dealing with aerobic respiration and oxygen. However, in anaerobic respiration, those electrons still need to be removed from the chain. Everything else is the same, but those electrons get picked up by something else. So for this example, sulfur, forming instead of water, hydrogen sulfide as a waste product. The waste products of anaerobic respiration completely depend on what molecule it is that picks up the electrons at the end of the electron transport chain. If you remember, we also formed two molecules of FADH2. FADH2 will also contribute to this process by dropping electrons off, only it drops them off at this second complex instead of the first. All this means for the cell is that a little bit less proton gradient is made because we skip the first complex and so we don't get to pump those protons. As you're looking at these, for every one molecule of NADH that drops its electrons off, we make about 3 molecules of ATP, whereas for every one molecule of FADH2, we make about 2 molecules of ATP. So if you remember from the last slide when we summarized everything, we had originally had 10 molecules of NADH. When those all drop their electrons off, they're going to lead to 30 ATP. We also had 2 FADH2, which will lead to another 4 ATP. When you combine the 30 ATP plus the 4 ATP here, plus the 4 ATP that were made through substrate level phosphorylation, you get the total 38 that you often see recorded in textbooks. This slide summarizes all the steps of aerobic cellular respiration, showing where the 38 total ATP would be generated. Remember that the number of ATP produced by ATP synthase is directly tied to the amount of proton gradient that was generated to power it. In anaerobic respiration, the electron transport chain doesn't generate as much of a gradient due to the differences in the terminal electron acceptor, so that value is a little bit lower for anaerobic respiration. This slide is included because it's a good summary slide that can help you study for the exams. I've highlighted the key difference for anaerobic respiration compared to aerobic respiration. This table gives some examples of organisms that use anaerobic electron transport chains and what their products are when they do. For example, Pseudomonas aeruginosa, which is normally a soil bacterium but can also be an opportunistic pathogen, especially in cystic fibrosis patients, are able to use nitrates as their terminal electron acceptor. When those pick up electrons, it's going to form nitrites. If this organism did not have nitrate, it would not be able to run the electron transport chain, and thus it would either die or it would have to rely on fermentation, depending on its other metabolic capabilities. I highlight this to make sure everyone realizes that not every organism will die if you take away oxygen, but instead they might be able to rely on other molecules the same way you rely on oxygen. Facultative anaerobes, which can use oxygen if it's present, but can switch to anaerobic metabolisms if it's absent, often have multiple electron transport systems, one that uses oxygen as a terminal electron acceptor if it's present, and another that uses something completely different as the acceptor if oxygen is absent. Alternatively, some facultative anaerobes do rely on fermentation in the absence of oxygen instead of performing anaerobic respiration. An important thing to note is that in bacteria, the proton gradient created by the electron transport system is not just used to power ATP synthase. In eukaryotes, that gradient is trapped within the mitochondria, and it's used exclusively by the ATP synthase within that mitochondria. But in bacteria, the proton gradient is generated outside the cell and can be used by any number of cell membrane proteins. While much of it is going to power ATP synthase, seen here, some of it is also commonly used in secondary active transport to either bring other molecules into the cell or to release waste products out of the cell. Additionally, it's the proton gradient that powers the motor that causes the flagellar rotation and movement of the flagella itself. These other uses of the proton motive force are one reason that the mathematical yield of ATP production is never actually met. So when you calculate that 38 ATP for cellular respiration, that's never actually met by the cell because the proton gradient is used for multiple other things, which means it doesn't all funnel through ATP synthase. And we also talked about how sometimes products are funneled off into different pathways and don't actually continue all the way through respiration. For both of these reasons, that mathematical yield is never actually seen inside the cell.